I would like to introduce Mike Dietz. He is from Boston University in the Department of Earth and Environment. Is that right? And I think, as Kenton kind of alluded, a previous collaborator of ours, and through some of those conversations, some of the ideas behind Brown Dog sort of were born. So I'll just hand it off to you, Mike. Thanks very much.
Thanks, Dave. Yeah, so I'm going to talk about the use case that we're doing within Brown Dog, which I guess is broadly defined on long-tail vegetation data. So this uncurated, unstructured data surrounding vegetation and how that's used in ecology and global change biology. If anyone you've ever dealt with ecological data, it is all 100% long-tailed, unstructured. I've never met anyone who's measured vegetation who didn't decide to store it in their own unique way. Even within a single lab, it will vary from like student to student and year to year. It's wonderful. So what my group is interested in is how climate change will impact terrestrial ecosystems. And broadly defined, we're interested in two questions. What is the strength of the terrestrial carbon sink, and how will that change over time? And how are ecological communities going to change in their structure and composition? This is a really important area in climate change research, because if you look, this graph shows the total emissions over the last few decades in red, and that partitions out into the fossil fuel emissions in gray, the land use emissions in black, and then the atmospheric increase. And you can see that all of these emissions factors are nice, smooth curves, but the actual amount of CO2 in the atmosphere, that increase on an annual basis is very variable, and it's considerably less than the emissions. And that difference is the ecosystem carbon sink. So half the CO2 emitted is taken up by either terrestrial or ocean ecosystems. And if you look at the global carbon cycle, you can see that the amount that's emitted anthropogenically is, it's sizable, but it's actually an order of magnitude less than, actually over an order of magnitude less than what the terrestrial ecosystems process on an annual basis. So what we're trying to do is detect, in understanding the change of the terrestrial carbon sink, understand the differences, the small net difference between two very large fluxes, and that little year-to-year variability in those two, the difference between what's going into the ecosystem and what's going out, is what's driving all of the interannual variability in atmospheric CO2. Now that interannual variability admittedly is often swamped by this overall trend of increase, which is a different problem. So the questions that I started with, where is the carbon sink going? What is the carbon sink going to be? How are ecosystems going to respond? Are questions about the future, questions about the current state. And so one of the ways that we address this is through the use of what we call terrestrial biosphere models. These are models that are trying to capture different aspects of the functioning of ecosystems, their biogeochemistry, their ecophysiology, the physics of the land surface, and the dynamics of vegetation communities. And they're trying to do this at scales ranging from individual trees up to the whole globe. Just briefly, since you aren't all ecologists, these include models that describe the pools and fluxes of carbon and other nutrients within the soil. This is just a quick schematic diagram of one classic model called Century that's been around for decades, describing how organic matter is processed in soils. These models are linked to models describing the physiology of individual trees. So GPP is the carbon coming in photosynthetically. All these other arrows within the tree are allocation and respiration terms. And then this blue arrow is the flux of water, and so all of our carbon fluxes are coupled to water fluxes. And then these models also represent what we call the biophysics of the land surface. so the hydrological side and the surface energy budget side. And then over longer time scales, these processes add up to longer-term dynamics of vegetation, such as these classic successional series of following disturbance, a predictable series of change in vegetation. Predictable under current conditions, maybe not as predictable as both the physiology of the species respond to climate and the climate changes drive changes in natural disturbances as well, such as changes in fires and insect outbreaks. Now the problem, or the challenge, is that when we take all of these pieces and put them together, each representing our best understanding how the ecosystems work, and we try to project those out over the future, this figure shows the land uptake predicted by an ensemble of different models. So each of those lines is a different model projected out to the end of the century. These are models run interactively with an ocean forced by ocean, so a fully dynamic ocean, fully dynamic atmosphere, fully dynamic land surface. And what happens, to put it briefly, is we have no freaking clue what's going to happen at the end of the century. That's the polite way of phrasing it. We cannot agree on the sign of the terrestrial ecosystem, let alone its magnitude. We have some ensembles saying that the land surface is going to be a source of carbon. We have some models saying it's going to be a sink of carbon. For reference, anthropogenic emissions right now are somewhere in the 8 to 10 range. So we're talking about potentially a sink equivalent or larger than current emissions by humans, or a source of similar size to emissions by humans. So that's a pretty big range of uncertainty. So how do we deal with that? How do we reduce that? So the idea is, the best way I can think of reducing this is to try to make the models better, and we do that by confronting models with data, bringing more data into the models. Today, I'm going to focus on two aspects of where we're looking at this. One, the PCAN project that Ken alluded to earlier, which is kind of looking from the present forward, and the other one, the Paleon project, which is looking to the past to understand vegetation dynamics. And actually, since I mentioned Paleon, I'll also highlight the interesting thing that most people miss in this figure, because you notice this big blow up on the one side. If you look back on the other side, you realize that the models believe that we have more confidence about the carbon cycle in 1850 than we do right now. So paleoecologists, we've got one in the room here. Great job. Good job. We understand what happened in No, we don't. That's an artifact as well. That's another screw up. Okay, so how do we reduce these screw ups? So challenges: explosion in the volume and diversity of data. As in most scientific disciplines, ecology and global change is in the era of big data. One of the challenges in ecosystems which have so many different interacting parts is that no single source of data provides a complete picture about what the terrestrial biosphere is doing. So we might have data from remote sensing, we might have data from inventory plates, we have data from towers measuring carbon fluxes, but none of them tells us the complete picture about what the carbon cycle is doing. In current analysis, you generally use only a small subset of those types of data, often the ones that are most available, most well processed, most cleaned up. And so I would argue that in many ways our ability to understand how ecosystems will respond to climate change is as limited, if not more, by the ability to use the data that we currently have as it is by the need to collect new data. We've also learned that the uncertainties in our projections with our models are as critical as the mean. If I had just shown you the mean from that ensemble, you would have falsely believed that we had a clue what we were doing. Another challenge being that when we do collect data, that is often driven by intuition rather than a real understanding of where the uncertainties lie. So PCAN was born as kind of alluded to, it's basically a workflow system to wrap around ecosystem models, to manage the flows of information in and out of the models, to automate analyses, and to make these tools more accessible to mainstream ecologists rather than just a subset of us who build these models. Pecan, started here at Illinois as a project that myself and David LeBauer started in the context of some work we were doing with Energy Bioscience Institute, and Pecan continues to be used in there, but it's also branched out to be an NSF-supported bioinformatics project that has collaborators here at Illinois, NCSA, BU, Wisconsin, and Brookhaven National Lab, so growing community. So one of the challenges we face in getting data into models is that we make measurements at hugely different spatial and temporal scales. And so what you can't do right now is just try to figure out how the data relate to each other directly. So we might have, a satellite measuring, everything, 1 kilometer pixels on the ground. We might have someone else going around with a chamber. So that thing up there, where you clamp that chamber to an individual leaf and you measure 2 square centimeters of the leaf. And this other guy's measuring 1 kilometer. And then you have these other guys over here pouring, liquid nitrogen into this thing they're about to drop into a lake that is measuring, pollen in a lake at 100-year resolutions to measure change in vegetation over the whole Holocene. And so how do you relate that to what's going on, in this tower down here, which is making measurements at 10 hertz? But all these processes tell us something about what is going on in ecosystems at very different spatial and temporal scales, all of which are important. So we envision the idea of models as scaffold rather than models as forward simulations, the idea that the model provides a way of linking different types of data together. Formally how we're doing this is we're trying to use data assimilation techniques. This is a quick and dirty cartoon of kind of our goal. So imagine we have an ensemble of models making different predictions about what's going on in an ecosystem. And so here on the left-hand panel, we might have, each of those dots might be a different model making different predictions, say, about the fraction of conifers on the landscape and the carbon flux across that landscape, and there might be some covariance between them. So these dashed lines in these other 2 panels are just what we call the priors. So the distribution, it's basically the projection of those dots onto their respective axes. So we have a fraction conifer and a carbon flux. And let's say we can measure one of those things easily. Say, in the present, we might have a satellite that tells us what the fraction of the conifer is on the landscape. 1,000, 2,000 years ago, we might have fossil pollen data that tells us what was the fraction conifer on the landscape. And so by combining the data and the observation and its uncertainty in black with the data and its the model and its uncertainty in the dashed line, what we get is the posterior, which is the synthesis of those 2 pieces of information. And the cool thing about data assimilation is that we then also get to update our understanding of the thing we didn't observe. So we constrain our estimate of the carbon flux based on its inherent covariance with the things that we did constrain, and that covariance is based on our understanding of the underlying processes. So one of the challenges there in all of what we're building here, there's many challenges, but one of them is managing these flows of information out. So we're trying to basically throw every type of data we can in the kitchen sink against these models because they're under-constrained. So we're building workflow. Pecan is kind of a workflow to handle this, but one of the bottlenecks, one of the challenges in all of this was getting data into the system to begin with. We had spent a number of years developing tools that were very good at automating analyses and analyzing the models and understanding their uncertainty of getting the doing that kind of getting the data into the model, but we weren't so good at getting the data into our system. So this is one of the ways that we're looking to work on the Brown Dog project of doing a better job of getting data into the project, into the system. And ultimately our goal is to make the tools that we're using of these modeling tools more accessible to the general community, so that running an ecosystem model is not a matter of going to a supercomputing center and learning how to bang away at the command prompt, as much as going on to a Google map and saying, I want to run my model right here, and then have the workflows handle the process of getting the inputs that you need off the web, doing all the conversions, doing all the extractions and processing that you need, and feeding those into the models in a very accessible way. So one thing to note, one thing that separates a lot of biology from other scientific disciplines is that in many other scientific disciplines, theory and modeling are often equivalent partners in the scientific endeavor with observation. In biology, they hate models. So modelers are a massive minority of the community, and so we are trying to ingest, we're probably outnumbered 100 to 1, trying to ingest all the wonderful data that everyone else is generating. So what we're trying to do is make these tools more accessible so that a larger fraction of the community can engage in this process. I will say the larger community hates models is a too strong a way to put it. They're skeptical of us. They don't know how to use the models, but they end every single paper they write or every single proposal they write with, these findings should be incorporated into these global change models. So we're like, okay, you're going to have to be equal partners on that. And so we want to make these tools accessible to the general community. So kind of our take home is that our models represent our understanding of how a system works. They can form this scaffold for data synthesis, and they're critical for forecasting how ecosystems respond. So improving models really needs to be a community effort. So this is why we are critically interested in ways to get all of this unstructured, uncurated data from the community into these systems in a more straightforward and more automated way. To give a quick example of kind of the stuff we're doing, kind of the test bed for where we're developing this right now is a region in northern Wisconsin, the Chequamegon Ecosystem Atmosphere Study, quite appropriately named by a bunch of folks from Wisconsin as cheese.
It's a good thing that they gave it that name instead of you.
Oh, no, they gave me that name long ago. In fact, I probably worked there for about 3 years before I learned how to pronounce Chequamegon. So I always just called it cheese. And here our goal is to reconcile information about the carbon cycle coming in from multiple data streams. So we have this region that has 17 towers measuring carbon flux, 250,000 tree inventory measurements, lots of ground measurements, and a whole slew of airborne or satellite remote sensing. And this assorted ground measurements is particularly lacking any structure or curation. So this is kind of what vegetation data looks like. I mean, this is the stuff we're dealing with. This is actually one of my notebooks that I pulled off the shelf in my office, and this is what the sort of data we're dealing with looks like. And this is how we're collecting it. I mean, people literally are walking around the woods with rulers and tape measures. And like I said, the people typically will enter this in some sort of spreadsheet, and what they name their columns are random and completely unrelated to what anyone else has named it before. We're also trying to deal with LiDAR data. This is an example of airborne LiDAR flown over one of our sites in Wisconsin. So this is airborne data measuring the height of the vegetation. So we get a profile of the ground, but we also get this kind of point cloud where there's vegetation. And from that, you can understand a lot about the structure of a forest. So where is the biomass? We're working with a Japanese satellite data called PALSAR that's a phased arrays synthetic aperture radar. So it turns out that the radar, since it has a larger wavelength, is actually particularly good at estimating or sensing biomass on the landscape. So this is an example of a biomass map inferred from radar data, but that also requires cross-calibration with all this on-the-ground vegetation data. We're working with hyperspectral remote sensing. So this is remote sensing that will measure So a typical camera measures, RGB. A typical satellite might measure 6 or 7 spectral bands. Hyperspectral remote sensing often will measure 200 to 400 bands. So basically, for every pixel, you get a full spectra. So this is very high dimensional data. It's flown at, it's been flown over our sites many times over many years. We're also working in a region in California where they're prototyping a satellite version of this that's going to launch, hopefully, fairly soon. Well, who knows with NASA. But from this, we learn a lot about, species composition. We learn about a lot of these physiological characteristics of the canopy itself that are important for understanding its responses. And really, the goal in our kind of northern Wisconsin test bed is to scale this up to being a system that can operate at a national or international scale. And a key component of this is the data that's about to come online from NEON over the next couple years. Brian, I don't know if anyone's heard of NEON. Brian interjected. He's our NEON person here today, and a few times asked some pointed questions. But this is an NSF-sponsored observatory network. 60 sites around the nation making the same measurements, using the same instruments, using the same protocols, which is unique to ecologists. We like to do everything uniquely. And potentially making something like, what is the current, last count I heard was like 527 different data products available, tens of terabytes a year of data coming on in real time about the state of ecosystems. So we're trying to build our system, knowing that we're going to get hit by this flood of data fairly soon. So I'm going to give one example of where we are now. So despite the fact that our use case was vegetation data, we actually started with weather data because we can't run our models without weather is the primary driver to the models that we're running. So we actually started there as the first input we needed. We're trying to build a system, using the brown dog tools, trying to ingest, anything from station data to these sort of regional reanalysis products that are themselves the result of these sort of data assimilation processes applied using atmospheric measurements and atmospheric models. We're trying to ingest real, weather forecast data so that we can do forecasting in real time, and as well as ingest the climate scenarios that are coming out of these global climate models that take us a century or more into the future. So right now we've kind of got a workflow that involves a number of conversions and extractions. So I think, as Ken alluded to, we like NetCDF right now, so we're using one metadata, one file format, and one file format NetCDF, and one metadata standard, the climate forecast metadata. So we're trying to take all of these different met files and formats out there, bring them into one common format. We can then, extract information from the regional products because we often only care about these on local scales. Do sort of various statistical analyses for downscaling and gap filling this data, and bring it into a kind of a unified product that leverages both the regional and local data. But then each of the different models in our system has its own quirky, unique way of wanting inputs formatted and structured. So we then have to do another set of conversions that takes these back out to what each individual model uses. And as complicated as that seems, it's a vast improvement over the current state of how models work, which is every single model out there has to figure out how to use every single different type of data. So by at least going to common formats and then branching back out again, we've dropped this from an order n squared to an order n operation. So that's kind of where we are now, and I've kind of highlighted the places where brown dog comes into this, because there's a lot of conversions and some extractions going on. I had to throw my little brown dog logo in there to make keep Kenton happy. So currently we're supporting at the fine scale met data coming in from Ameriflux, which are these towers making carbon flux measurements from a series of elevated CO2 experiments. And then we've also set up a system where we can ingest CSV formatted data as long as the format, what the headers are in that data are registered with our system. So one of the things we've been talking about with the Brown Dog team is, can we use the data tilling service that kind of does metadata extraction to fill in this understanding of the formats of these data on the fly, so that we don't have to sit there by hand every time we come across new metadata and go, okay, they used air T instead of T air for air temperature, so I need to figure out what column that goes in. It's just a pain. It's time consuming, and it's one of those things that forms a bottleneck that prevents us from using data. And so and then at a regional scale, we're just currently using one data product, the North American Regional Reanalysis. Though the regional ones are actually much simpler, because from this perspective, from an infrastructure, from a coding perspective, they are much more predictable in their formats, much more unified. It's all the site level data that's all over the place. So we are working towards, again, supporting the core of our, there it's back, kind of the core of our case study is to get to the vegetation data fairly soon, where we have collections of different types of data that give us different pieces of information, sometimes about the initial conditions, what's the current structure out there right now, and then as well as the dynamics of the system. So this is kind of showing some of the types of data we want to ingest and what we learn from those sorts of data. So it's different types of remote sensing, like the LiDAR and radar tells us about structure, the hyperspectral about composition, Landsat tells us about land cover, and then repeat measurements tell us about the demography, phenology, disturbance, and general change. And so we kind of envisioned this general workflow of, again, of any, using the DAP to distill data, the variety of data out there into a standard that then allows the PCAN tools to build all of our tools so we always act on one standard, to process this and then eventually convert those back to model-specific values. That's part one. So let's come back here, and part two is to think about looking backwards at this paleoecological data. So Paleon is a fairly large NSF macrosystems project. There's over 60 people involved. It's headed by folks at Notre Dame. And what we're trying to do is use historical data about vegetation and prehistorical data about vegetation to calibrate initially just to validate these land surface models. It's interesting. We built these models to make projections out at a century time scale, and then we've only ever validated them using contemporary data on a decadal time scale. So we were like, maybe we should, validate them at a centennial time scale. But that requires finding the only available data about that is this paleoecological data. So compiling that data, and then trying to use that to make inferences about what was going on during the pre-settlement period, and how, not to get in the weeds here, but one of the reasons that we had this false impression here that we were more certain about 1850 than we are at the present is that we assumed that reality was in steady state at 1850, but it clearly wasn't. And so the initialization task is just saying, if we drive the models dynamically with historical observations, how does that change? So what happens when we get rid of that equilibrium assumption? And so we're specifically going to focus on this last 2,000-year period, and these are kind of showing some of the climate signals that have happened there. There's this medieval warm period and this little ice age. And then all of that is swamped by current warming. The larger goal here is to bring a whole bunch of different models together. That's what's on the acronyms on the top row, with a whole bunch of different data. That's the stuff on the back row, the bottom row, in order to do this. And I'm going to focus here today just on one little part of this, what we call the witness tree data. So this is data that's coming from these handwritten surveys at the time of settlement. So I don't know how many of you are all in the Midwest now. I don't know. And if you've been here a while, you will notice that everything around you is laid out in 1 mile grids. from here to the Pacific, everything is 1 mile grids. That 1 mile grid was established during the settlement era, during the surveying. What most people don't know is that at the intersection of those every 1 mile grids, they've actually measured the vegetation, in the 1850s, 1860s, and they recorded it in these hand-rated notes. It's actually a higher data density about vegetation from that era than we have about the present. The problem being it's handwritten. But so this is, Ken didn't talk about his census project that they did. Luigi will. So there's a lot of analogies between this and a project that Luigi will highlight later. But the idea is to try to extract the vegetation data from these handwritten records that are often, scanned. Some of them are in PDF, some of them are in image format, some of them are in microfiche, some of them are still in paper records that we'll have to go scan. But then to use that to learn about what the historical state of the ecosystems were. So these are some results that we've done synthesizing the existing data just for the upper Midwest. And it should be noted that that effort of filling in that little grid there from all these handwritten records was done by hand by undergraduates reading those records and making notes and entering them into a database. It took the labs involved about 25 years to generate that map. That's not a scalable solution. We're hoping to accelerate that a bit. So that's kind of the second half of our case study is to try to dive into those. And this is kind of showing, we can find out what the species composition is. We can find out a lot about the structure in basal area and the carbon pools in this era before human settlement using this sort of data. And this is a critical baseline to judge human, in many ways, not just climate change, but conservation, just the basic baseline to which we judge anthropogenic impacts against. So that's all I had, unless there were questions. Yeah.
It's not ecology, but I'm from geology.
Yeah.
And so, am I on now? Okay.
Yeah.
So we have a lot of handwritten maps as well, long before GPS, right? And then three-dimensional reconstructions, those are interpretations. And our pollen data, we go back to Mesozoic, and other fossils that your scenarios may sound horrible, but for geologists, it's order of magnitude worse.
Well, you have to deal with orders of magnitudes more time, and the data just gets messier
so
and messier.
If we can get data, VARB data, we can maybe get annual resolution from lake sediments, but 1,000-year resolution, 2,000-year, 10,000-year resolution, or sometimes even million-year resolution is best we can hope for. And we may have a precise chronology, but it's floating, right?
Yeah.
So how to reconstruct past geologic history becomes much more daunting than what you're faced with.
There's direct analogies between that and what we do with the pollen data as well, with the floating chronologies and course or time resolution. I just highlighted one
I was surprised
example.
to hear you say pollen data, because we always thought it was our domain. We use pollen data a lot for paleoclimate reconstruction, which is primarily, I thought, was being done in earth science domain rather than because we go back.
The thing that pollen comes from plants.
I know. I understand. But I use pollen, I use ostracods, diatoms, anything, and it's all living things.
Yes.
But without earth, the solid earth as the substrate, biology doesn't exist.
Yes. Yeah, Brian.
So Mike, Brian Wee from Neon, for those over the ether. Quick question again, kind of minor in detail.
Sure.
Do you know if the historical derivations of species distributions when that information is derived, is that information submitted to the Global Biodiversity Information Facility, or GBIF, based in Copenhagen, as almost like a museum record might be submitted to GBIF?
To my knowledge, that has not. So the pollen data that we use has its own kind of project called Neotoma. That's kind of the repository for Holocene period fossil pollen data. That's an international collaboration, probably another NSF-sponsored project. I don't know if they've ever thought about linking that to GBIF. I'll bring that up with them next time I talk to them. The witness tree data is an interesting story in that there's kind of, because of the labor that went into it, the labs that generated this have put restrictions on our ability to redistribute that data. And the papers analyzing it at this larger scale have just been submitted, so I do not personally know what the plans are for what specifically will be released from that. I know the postdoc on that project is a huge advocate of reproducible science, so I suspect he's going to make as early in his workflow available as he is allowed. He wrote all those manuscripts in our Markdown, so he can recompile them. Yeah, so we are very conscious of those sort of data sharing issues, but I don't think any of us had thought specifically about putting that into GBIF. That's a great idea.
So if there's one other question, maybe Mike, if you could step away from the
Sure.
podium, and we'll get Praveen and Gina, if you want to, or whoever's coming first. So let me just invite our next speaker to get technically set up, and then Mike, maybe you could answer a couple questions while we get set up at the podium. So.
Well, I actually had a quick comment based following up on Brian Weiss and actually Emmy Ito's sort of turf discussion about paleoecology, being a palynologist and working on pollen. I think what it gets the heart at, though, is like some of the interdisciplinary sciences that live in two worlds. We actually do deal with multiple data formats, right, and multiple timescales, spatial issues. And I think the power of something like this, right, or approach like this, an integrative approach that crosses disciplinary boundaries is really, really critical, right? So when we work with data sets, so it's not whether or not data resides in geology or biology, or I don't think that's of a significance, right? Being able to integrate between fields is probably.
Oh, I agree completely. I apologize for being a little snarky there. And yeah, when I was first introduced, my home department is an Earth and Environment department that ranges all the way from mantle geophysicists to environmental economists. So I'm well aware that we all depend on each other academically to.
Yeah, but integrating between something like Neotoma and GBIF is an issue because, of course, GBIF doesn't think temporally, right? So it's only a spatial database, whereas Neotoma is both spatial and temporal.
I don't have an option.
But the issues of taxonomy and consistency of taxonomy, which you get in GBIF, right, the updating of taxonomy, doesn't happen in something like Neotoma, which is an issue, right, because there are taxonomic revisions occurring all the time, and so the integration of phylogenetic information actually doesn't occur in these geological data sets or easily. So.
Okay. I'm not 100% sure. I think Neotoma may actually be working in that direction. I think
really
they have taken a lot of thought into the evolving taxonomy problem. But so it may be working one way, but not the other.
Okay.
I'm not intimately integrated with Neotoma to know, but.
Okay.
Let's do one more question here, and then we'll get started. Yep.
Well, I have more of a comment too. I think Mike said something that really resonated. In your overview, Brown Dog overview workflow, you showed, the analysis happened in the computer and then it was straight to publication. And what you said was this data is evidence. And in a humanities discipline, that is the key, that, the data is not where the analysis takes place, or the human has to interpret, and we use it as evidence. And I think that that's key too to the kind of issue of not getting credit for tenure for creating applications, because if you can show that the analysis or the interpretation either went into creating the algorithms or into interpreting the data that comes out of the computer, then that really makes a stronger case for, you know, the value of kind of this kind of work. So that really resonated, especially when you talked about predictive modeling. No one knows what's actually going to happen, so you have to use this data as evidence and then make your best guess, but your interpretation is more important. So it's just kind of a comment, and I really appreciated that
Thanks.
emphasis.